So welcome back, everybody. We're narrowing down to the end of the semester. Anyone have anything they want to bring up to start with? We had a lot of things that are due today, right? Discussion boards, there are 3 discussion boards due, and then make sure homework 8 and 9 are getting in, right? No questions on homework 8 or 9? We good? Okay, we're gonna switch gears today. We are gonna go into our last type of model, generalized linear mixed effects model. And we're gonna do that for 2 weeks, and then we'll wrap up with the sample size and other problems that can crop up. So let's see, I think that this PowerPoint that I'm gonna go through, which is actually one of Jameson's old one, is in the Files tab under lecture. Let's see, is it? Yeah, it's Files, and then under Lectures, and we're down here to lecture, what is it? 10 GLMM. And it's, let's see, what do we have in here? Yeah, there's that. This lecture 10 PowerPoint, that's what I'm going to start today. We're not going to get through the whole thing today, but this is where we're going to start. Okay, so I'm going to, let's see, is it going to let me, pointer options, laser pointer. Haha, I got my laser pointer out. Okay, I'm going to have to shrink you guys' pictures, so I'm not going to see if you do the raise your hand button or anything, and I'm not going to be able to see your faces very well. So if you have questions, please speak up, because that's pretty much the only way I'll be able to tell, okay? So today we're going to talk about these generalized multi-level models specifically for binary outcomes. So that's like logistic multi-level modeling. You can also do these for Poisson and count. So today we're just going to do slides, and then we'll first part of the slides, and then we'll do the next class day, we'll do some more slides and more examples. So today we're not going to do a lot of working in our examples, but we will soon. Right now, I don't have any homework listed. A lot of times I don't even assign any homework for this topic because you're working on your course project so much. I will probably put up a discussion board with some information for you guys to read, some journal articles and maybe a video to watch, but I don't think I'm going to assign a homework like an R homework assignment that you do on your own unless you really want one.
Thank you.
No, nobody's going to fight for another homework assignment, are they? Yeah, I can if you want one.
So does that mean we should just ignore the homework 10 that's on Canvas?
Yeah, so homework 10 and homework 11, I'll probably end up deleting. I've left them there as placeholders, but I don't have anything prepared to send out with them.
I was hoping we could do something with power analysis though in MLM.
Yeah, we will do something with power analysis. It'll probably be more interactive and less like homework assignment-y. And the reason is I don't want to overtax you at the end of the semester when your course projects should be your primary focus. But I will give you options. We'll go through examples and the tools that are out there to do power analysis for multi-level modeling, because that is, once you understand them, that is the hardest thing about using them, is to figure out what your sample size should be beforehand. And it's a big deal if you're going to write a grant or write a proposal. Okay, so here we go. So we've already seen logistic regression for single level and using a GEE. So logistic regression is when we have our outcome that is a 2-level variable. It's sometimes a yes or no, sometimes it's presence absence, but the idea is we have 2 levels, and we usually code them 0 and 1. 1 is the thing we're interested in, but you can label whichever one you want, but we usually call them 0 and 1 for a condition, a state. So when we do that, the distribution we use is binomial or Bernoulli. Bernoulli is if you only have one time. If you have multiple, we call it the binomial. So if you roll the dice one time and you want to know if you have an even number, or you flip a coin and you want to know if you get heads versus tail, one time would be an Bernoulli, but if you do it multiple times, it's the binomial distribution. Bi meaning 2, nomial means multiple times. So we're going to use the binomial distribution with the link equals logit, the same way we did with single level regression and GEE, but we're going to do it in the multi-level or mixed effects framework. So we're going to go back to having random effects. So in the last 2 weeks, we've talked about GEEs. So GEEs you can do with the normal Gaussian distribution, or you can do it logistic, Poisson, gamma, all these other negative binomial distributions. When we did multi-level models, we did fixed effects, random effects. The random effects were to count for the lack of independence. When we did a GEE, instead of having random effects, we used a correlation structure. So that's the main difference between a multi-level model, whether it's an MLM or a GLMM, and a GEE model, is for the lack of independence, do you use random effects? Or do you do a correlation structure? The fixed effects are handled the same way. When we do random effects, it's maximum likelihood based. That's why we can do the likelihood ratio test. When we do GEE, instead of doing random effects, we do the correlation structure, like the autoregressive, the exchangeable, the toplets, all those different ones. And it has to fit it not with maximum likelihood, but with an iterative or an over and over cycling process. So we're going to go back to the mixed effects with maximum likelihood. Okay, so we're going to focus first on longitudinal binary data. Now, these slides were written by Jameson Fargo, and I think that the first time he made these slides were like 10 years ago or more. So they are slightly outdated. So he does say on here, the first bullet point is that modeling longitudinal binary data is currently extremely complex. Now, 10 years ago, even 5 years ago, SPSS could not do this. It wasn't even an option in SPSS. SPSS could do multi-level models, but they couldn't do generalized multi-level models. In fact, I was one of my first consultations here 5 years ago was somebody submitted a proposal. I can't remember if it was a grant or a student proposal, and they were going to do logistic multi-level models in SPSS. And I think it was a committee member said, you can't do that in SPSS. But since that person had tried last time, it had been introduced. Now we can do them, but the second bullet point is still in effect. Convergence can be a problem. Now, it used to be that it took a long time to make these converge. Like you would sometimes have to let these run for hours. With our computers nowadays, it doesn't take as long, but you can still have convergence issues. So because of this, GEEs are sometimes used instead of GLMMs because convergence is less of an issue. Now there's a con to go along with that pro, and we'll get to that on a slide coming up in a couple slides. I want to go, oh, this is slide 23. I didn't go all the way back to the beginning, did I? I think we need to go back even further. Yeah, okay, we'll go back even further. This is better. I didn't start on slide 1. You guys are looking at me confused. I'm sorry. Okay, let's get that little pointer thing back. Okay. So the GLMMs, it's like Jameson put, they're relatively new, but new is like in the 90s. So at this point, they've been around for 30 years. So again, back in 2000, when these slides were probably being produced, they were brand new, like as far as being used by mathematicians developed these first, like statistical mathematician theorists, and then they start coming into being applied in different areas. So we're going to combine the idea of generalization with the random effects. So because we have mixed effects and the generalizations, they are more complex alternatives to the GEE. And again, in GEE, how do we handle lack of independence? With a correlation structure, versus in mixed models, we use random effects. So they're similar to multi-level models. It is a multi-level model. We are going to have fixed effects that we assume to vary randomly across clusters. And the random effects are to deal with everything else that we didn't measure. So random effects pick up all of the person-to-person variance that is not attributable to things we measured. Now, somebody had this in a discussion board about that had a question about, and I think it was over the GEEs, that the model uses the fixed effects to predict or to model the mean values. Does that sound familiar to anyone? No? Okay, we'll come back to that a little bit when we see an example, but we're still going to have fixed effects, and now we're going to have random effects, but we're going to have this family. So we're still assuming that the highest level clusters, the highest level units are independent, but within each higher level, we have a degree of correlation. So they're the same as multi-level models we've done before, but they are slightly different. So the random effects are unlikely to be normally distributed. So remember with MLM, we did residual diagnostics at the end, and we wanted to see a normal curve if we looked at the histogram of the residuals, and we wanted to see it level above and below the line if we looked at the residuals. That is unlikely to be the case in the generalized situation. For instance, if we are in a logistic regression, 0 or 1, our model is going to predict 0 or 1, and the truth is either 0 or 1. So the residual is either 0 or 1. There's no way that can be normally distributed. Another reason we'd use this is if the predicted values are within a restricted range or bounded. Like logistic regression, we only have 2 outcomes. We don't want to have a predicted value of 3 if it has to be 0 or 1. If we're doing a count that's 0, 1, 2, 3, 4, 5, we don't want a predicted value of -7. That makes no sense. So we're using these other distributions usually to rein in the bounds of our predicted outcome, as well as the shape of the residuals. So here, okay, this will be good. This is the one that talks about the mean value. So there's 3 parts of these models. These 3 parts should look familiar. The first part is the distribution of the outcome on, we have some family. So we're going to say a family. Binomial with the logit link to do logistic regression. Poisson with a log link if we have a count. And then this number 2, this is the one I was trying to find the words for. The conditional mean of y is assumed to be dependent on fixed and random effects and is linearly related to the fixed effects via the link function. So this is saying that we can use the fixed effects to predict the average value of all people that have those fixed values. We have the covariates. We assume the random effects are independent of the covariates. Okay, I skipped a slide, didn't I? Okay, no. Okay. Another way to think about these models is that the random effects, and you can redo, any multi-level model can be redone as a structural equation model where the slopes and intercepts, instead of being random variables, are unobserved latent variables. But either way you do it, the assumption is that the random intercepts and random slopes, those are normally distributed. Okay. Okay, for computational convenience and simplicity, random effects are typically expressed in standardized form. Yeah, okay. I want to get us to, there's a slide that has some pictures. Okay, betas. What do the betas tell us? The influence a covariate has on a subject or cluster's mean value of the response. And how do we interpret betas? It's the effect of increasing the X value by 1, holding all other covariates constant. And it's not just holding the covariates constant, it's holding the random effects fixed or constant. So interpretation-wise, the parameter estimates, because of using random effects, our beta parameter estimates are subject-specific. When we do GEE, they're population-specific. And I want to get to the next, I think it's the next slide that has some pictures. I'm going to go to that picture slide. Where is it? Okay, I want to look at this. Okay, so let's look at the top two pictures first. So this first one is, we're looking at these first top two pictures are if we have a continuous outcome. So X, this is a repeated measure scenario where we have repeated measurements on people. So the X axis is our main predictor, time. So we're saying what happens over time to the outcome, that's Y. So in this MLM, we see that over time, what happens to our outcome, it goes up on average. Now these little dotted lines there's like one for each subject. It's only showing about 4. So does every subject start the same? No. So this would be the random intercepts. Does every subject increase at the same rate? No. It's a little harder to see. Some of these, are they parallel? Look at the gaps in the lines. The gaps are growing over time. So that means the slopes have to be somewhat different. So this picture of the generalized MLM analysis is showing that over time, we have random intercepts and slopes, but there is a general trajectory for all people to increase over time. Some people start lower and increase slowly. Some people start higher and increase more quickly, but over time, all people are increasing. And this dark line is the fixed effect line. So that shows on average how fast people are going up. Each of the dotted lines combine the fixed and random effects. So that's the BLUP, the best linear unbiased predictor for each person. Now, I want you to compare that to the GEE version. Now, this is with a continuous outcome. Check those two pictures. How do they compare to each other? The MLM and the GEE.
I think the GEE, it seems like they widen quicker, I think.
You think? They should look identical. It's hard because we're seeing the wide ends compared to the narrow ends. But if you were to put them on top of each other, they should be identical. Now, the thing is, with the GEE analysis, you only ever get out this dark line. You cannot get out these dotted lines. But the dark line for the parameter estimates in GEE will be identical to the parameter estimates in MLM, assuming you pick the right correlation structure, or good enough, and the right random effects for MLM. So for continuous outcomes, GEE and MLM give you pretty much identical results. So if you have a continuous outcome, MLM is better than GEE, but they will give you the same results. Now I want you to look at the bottom two pictures. Do these look the same? This is doing logistic regression. So the chance of something, maybe it's the chance of graduation. So over time, what happens to the chance of graduation? The graduation rate. So let's look at the GLMM on the right-hand side first. So each of those curves is for a different person or a different kind of person. Maybe it's for males, females, different ages. Maybe they're young. I don't know. We got a curve for each person. Notice the dark curve. How does this dark curve compare to the dotted curves?
Parallel.
It's parallel. It's the same. It's the exact same shape. Now I want you to look at the GEE version on the left-hand side. Now compare this dark line to the dotted lines.
Maybe it's like the average of all the lines.
It is the average of all the lines. Julie hit it on the nose. GEE tells you the population average averaged over all people, where the GLMM says for an average person. The words are very close. Let me say it again. The the mixed effects model tells you for an average person. The focus is on the person. GEE says averaged over all people. So within the population of all people, the average does acts this way. The words are slightly the same. What happens is if you have a continuous outcome on the top, those words reveal exactly the same mathematical picture. But looking down at the bottom, if you're generalizing to a non-normally distributed outcome, there is a subtle difference. So you can fit, if you have a binary outcome with cluster data or repeated measures, you can fit a GLMM, a generalized linear mixed effect model, or a GEE. But it depends, your choice of which should depend on how you want to interpret your results. Do you want to interpret it based on how a given subject will behave, or how the average of all subjects behave? Now, if I am a teacher, what am I more concerned about? Individual people or the population average of all people, students? If I am a teacher.
Probably individual.
Probably individual kiddos in my class. If I am the state education superintendent, what am I more concerned with? Because I'm making policy calls. I'm more concerned with the average of all. Now, often I ask researchers what they are most concerned about, and guess what they tell me? What are you most concerned with?
The individual.
Individual, or they say both. So because of that reason, the GLMM usually fits most researchers' goals better. However, that's the bonus. You can interpret it the way you want, subject specific. The problem is it fails to converge more often. And so you can't even fit it. So a GEE is like our best way to get a hold of the data. Now, in both of these bottom pictures, over time, what happens to the chance of graduation? Over time, in either of these pictures, over time, graduation rates increase. The shape is different, so the predicted values in the middle will be different, but the ends are the same. The message that with time, we have an increase in this outcome remains the same. So often the GEE and the GLMM will tell similar stories, but they will be slightly different in the interpretations, and the betas will turn out, and the exponentiated betas will be similar, but not the same. I'm going to go back a couple slides. Okay, to this, let's see this slide. Okay. So here is generalized multi-level models are computationally challenging. We can do them with our computers now and our software. We can. We have the ability to. They don't take a long time to fit, but sometimes they crash and burn still. And the reason is because of the complexity of the models. So, well, here's an illustration. So let's really simplify this down. Let's talk about if I only have 3 people in my study, person A, person B, person C. And we have 2 time points. That's about as simple as we can get, right? So here we're looking at a binary outcome. So just 2 time points. So here's person A, time point 1, time point 2. What's the difference? They went down. Person B, 0.5, 0.33, they also went down. Person C, 0.2, 0.11. Everybody's going down. The average of the averages is the same as the average of these averages. Have you ever done a table where you did the averages going down, the averages going across, but then when you average both margins, you end up with the same thing? It's just which margin we want to focus on, the average of subjects or the average across time. And we're still going to do that log odds ratio thing. Okay, example time. Let's think about a cross-sectional example. Cross-sectional is where you only have 1 observation per person, okay? So what's the lowest level? People. So here we're going to do kids. So we're going to talk about, think about kids in different schools. What's level 1? The sample of all kids. What's level 2? The sample of all schools. So we have schools, and within each school we have kids. So consider the predicted value for 2 male students from different schools, but their schools have the same SES. So we have 2 boys, one at this school, one at that school. Their schools have the same SES. Should they have the same predicted value? With a population average, with a GEE model, the individual schools are not of interest because we're just using a correlation matrix. So it would predict values would be the same for both students. The GLMM would give unique values, would incorporate the uniqueness of the schools beyond just having the same SES. So we'd get slightly different predictions for each of the 2 boys, because it's going to be specific to each school, not averaged over all schools. Okay. Here's a longitudinal example. So consider we have 2 boys measured over time. So now we're not, ignore schools. We don't have schools anymore. They're just 2 boys over time. So what's level 1?
Time measurement.
Level 1 is the observation times. How are they grouped together? By being from the same person. So now person is level 2 and the observations are level 1. So we got 2 boys being measured over time. The population average model, the effect of individual trajectory is not of interest. Where those 2 boys, their predicted values would be the same. Whereas with a random effects model, the 2 boys would get different predicted values because they would have unique random effects. I can't get these pictures to shrink down again. Now they're all big. Where are they? Okay. If your interest is in making population average interpretations, go with GEE. If you're interested in how changes in a fixed effect relate to individual clusters or units or people, then you want to go with the GLMM. I can't, there we go. Okay, with GLMMs, we're going to do a model fitting procedure and testing just the same we did with MLM. Now, usually what kind of model do we start with? A null model, right? With just nothing in it. For a GLMM, the first thing we want to add are random effects. So we're going to always start with random intercepts. We might check random slopes. And then we're going to add fixed effects. Usually I do this in a very similar manner to MLM. It's just GLMM. Here's the good news. In R, the syntax is going to be nearly identical. What command did we use to fit an MLM?
LMER.
LMER. For a generalized multi-level model, you just add a G in front. It's GLMER. The formula is the same, the data equals is the same, but we have to add family equals binomial with the link equals logit. Other than that, it's the same. We're still going to fit nested models, making them more complex, and comparing the model. Since we're back to mixed effects with maximum likelihood, we can compare models when they're nested with each other with that ANOVA command. We liked that, right? That was a fun, maybe not fun. That was a nice clear-cut way to decide if a model was better. So that's an advantage over GEE, is it's more clear on comparing nested models because we're back to using maximum likelihood. Now, here's another difference. What was another thing we had to worry about with mixed effects models, multi-level models? When we use lemur, we had to give it a formula, we had to give it a data set, and what was the last thing we specified? that was always a pain to figure out which one we were doing.
Remel versus ML.
Remel versus ML. So here's another piece of good news. You can't do Remel with the GLMM. So you don't have to do Remel equals true. You can't do Remels equals false. You don't have to worry about switching back and forth. There's no such thing as Remel for a generalized linear mixed effects model. Whew, that's one less thing to worry about, right? Okay, diagnostics. Not well studied. This is I have a sad face. We still haven't figured out really well how to do diagnostics on GLMMs. Not very well figured out. We do want to check the random effects, but it's highly likely our random effects are not normal. And that's just the way it is. We just have to deal with it. So you can look for outliers the same way, but generally we just don't do diagnostics for GLMMs because they're not well understood. Just like how often do you do residual diagnostics on regular linear regression? We never do. Okay, so Jameson had this slide in here with all the different programs you can use. So notice, what does it say the very last bullet? Not SPSS, because SPSS used to not be able to do these, but now I think it was SPSS 23 or so. We're on now to SPSS 26. SPSS can do these, but I would tell you they are clunky as all can be. Avoid SPSS at all costs for this type of model because you will want to throw the computer across the room. I've done it. Didn't go well. So notice what the very first bullet, this is what we're going to use, LME4. Now, when we were fitting our lemur models, we ended up using LMER test package. Do you remember why we used the LMER test package? What was the benefit of that? Anyone remember? No? It's because it gave us our wall tests. Ah, I'm glad we got some recognition there. You cannot do that with Gleamer. You have to go use the LME4 package. You can't get wall tests. The LMER test package still does not have the availability to do Gleamer models. So we do have to use the LME4 package. So here's where Jameson says that sometimes they run for several hours. My experience has been now they don't run for hours. You know within like 2 minutes whether or not it's going to crash and burn or it's going to run. So that's good news. Something's gotten better. Yeah, so this last bullet, it's not that new anymore. We're pretty good with them. Okay, so here would be some code. So we're going to use the Gleamer function from the LME4 package. You're going to put your dependent variable, tilde wave, any independent variables, plus random either intercepts and slopes per cluster variable. That look familiar? Yeah. But now we have to add family equals binomial, link equals logit, or Poisson, link equals log, or whatever we're doing. And data set equals, you don't have to do Remel. Sometimes we add a different method. So there are different methods to fit these. And I think Hawks talks about them some more, and we'll talk about them more next time. I'll put the reading up for a discussion board that we can read over the next week. But the three main methods are PQL, Laplace, and AGQ. We'll talk about those more next time, but there are some, we don't have to do Remel, but these methods are like, and sometimes we have to do the different optimizer methods. Okay. Let us take an early break, shall we? Does that sound good? Okay. Let's take a 5-minute break. Let's come out back at what, 5:12-ish. Allison, yay, got a thumbs up.
when it gives you the EM mean value, what is that value?
Estimated marginal mean. So that's like the predicted value for the average random effect. Was that Greek? So that would be like the dark line on our pictures we were just looking at.
Oh, yeah. Okay. That's the actual.
Mhm.
Because that EM means, I'm using that EM means to try to get some significance values and things. And
Yeah, using the
Will
EM means is nice to get predicted values on which you can do like post hoc testing, or those are the points that are plotted. We have some nicer plotting tools now that are easier, but yeah.
does it show if it's significant? Does it show stars usually?
Well, you have to, after you calculate the estimated marginal means, then you have to do comparisons on them, and that will give you stars.
Okay, good. I'm not starless yet.
Oh, I should probably pause our recording. Okay. I thought it would take us longer to go through those slides, but I'm ready for an example. What do you guys think? Should we do an example? Okay. This is going to be a fun example. Not, sorry. Especially for the guys, but oh well. This is the data set that is one of the traditionally used data sets to do GLMMs, generalized linear mixed effects models, the first time. So we have to have an outcome that has two levels, yes, no. So this is data on amenorrhea from a clinical trial on contraceptives for women. Amenorrhea, which I might be saying wrong, is cessation of menstruation. So contraceptives are birth control. So these are women who are taking birth control, and often a side effect of birth control is the hormones cause a woman's monthly cycle to stop so she doesn't bleed monthly. So this is a longitudinal study. It was a clinical trial. So they randomized women to either get an injection of either the low or the high dose of the contraception. And then they followed up, they had an initial injection and then 3 additional injections. So they had 4 of these birth control injections 90 days apart. And at each time they asked them if they had, I think they had them do a journal. Is that what it said? They had them do diary data. So they keep track of their cycle and note when they stopped menstruating, if they did. So our outcome here is, yes, they stopped. No, it's still going on, right? So it's a yes or no outcome at 4 time points. So we've got the first time and then these follow-ups. So we guess what happens with our 1,000 and some odd women over time? Do all 1,000 plus women finish the study? No. They don't. Now, we could not do a repeated measures here because repeated measures ANOVA. Because with repeated measures ANOVA, you have to have a continuous outcome. Our outcome here is not continuous. It's either yes or no, 2 levels. So we can't do repeated measures ANOVA. So you have to do a repeated measures either GEE or a GLMM. And today we're going to do the GLMM, generalized linear mixed effects models. So we've got these 4 time points that are each 90 days apart or 3 months apart. So our variables, we have an ID variable to uniquely identify each woman. We have the occasion variable, so occasion 1, 2, 3, 4. And then we have the outcome status, which is either, yep, they missed their period, woohoo, or no, they're still bleeding. And then the randomized variable is whether or not they were given the low or the high dose of the birth control. So we want to know if over time, does the high dose lead to a bigger chance of having the cessation of their menstruation, or are the 2 groups, the 2 levels of dose, the same over time? So we have repeated measures, but an outcome that's 0 or 1. So here I pulled the data in. It's very small, like only 4 variables: ID, dose, occasion, and the outcome. When I pulled it in from, this was in their, on their website, it's a text file, TXT. So text files you can read in very easily in R. You just do read.table. This is a comma separated text file. So if I ask, so like here's woman number 1 in the study was on the low dose, and at, let's see, how many times did we get her to come back? We have baseline and nothing else for that first woman. Lovely. What about the second woman? Baseline and nothing else. But here's the last person in the data set. They came all 4 ways. We got them to come back, and they were skipping their period every single time. So this is in long format. I decided now the way the data set came in, ID is an integer. I want it to be a factor. Dose is either 0 or 1. I want to label them as low dose or high dose. Occasion is 1, 2, 3, or 4. What do we think about occasion being 1, 2, 3, 4? Is that how we want it? What might be a better way? Start with 0, so subtract 1. And then the amenorrhea variable is coming in as a character, which means it's text, so we want to make that a numeric. Always, if your outcome is 0, 1, it's best if you recode it as the number 0 and the number 1 and not as a factor. It causes less, R is getting better, but sometimes it can cause problems. So that's what I did here. I made the ID variable a factor. For the dose variable, instead of being 0 and 1, I labeled them low and high. And on the occasion, I subtracted 1. So instead of going 1, 2, 3, 4, it starts with 0, 1, 2, 3. And then the amenorrhea, I had to make it as a numeric value. And I only wanted to keep complete lines, like only the follow-up data where the woman actually came back. So this is how the data set looks like now. So person number 1 only has occasion 1, which is time 0. Occasion is the 1, 2, 3, 4. Time starts with 0, 1, 2, 3. What about person number 4? Yeah, okay. What about person 1149? They came twice. So here's all our people. And then I made it to wide form to get us some descriptives. So let's do some visualization. So visualizations are a little harder when you have binary 2-level outcomes. You can't do a traditional spaghetti plot because everyone would be at 0 or at 1, and you just have lines connecting 0s and 1s. So a better visualization is looking at the percentages at the different times. So this is at the first time when they initially come in, those, how many people are observed to already have amenorrhea at the first visit in the low dose and the high dose. And this is randomized. And then each successive time period, so these are 90 days, they only saw them every 3 months. What happens to the chance of amenorrhea over time? The chance of experiencing an amenorrhea goes up. Looking at this plot, can you see a clear difference between the low dose and the high dose? Who's more likely to have amenorrhea? Which column's taller?
High dose.
High dose experiences the outcome more, but is it constantly more, or does that gap widen or shrink over time? It's hard to tell here. That's what we want to address. It's also hard to address here because the sample size is changing. So at the first time point, we have over 1,000 women, but by the end, we have fewer, substantially fewer. And so we want to control for that. Here is the same data, but I've connected the lines. Instead of doing bars, I did a dot at the height of the bar and then connected the dots. Makes it a little easier to look at. So the red line or the high dose is above, but are they parallel? Ish. Okay, so here we're going to fit our first model. We're going to use the LME4 package, Gleamer. So our outcome is that 0, no, 1, yes, amenorrhea variable. And we want to know over time, is there a difference by group? Does dose moderate the chance over time? And this one has only random intercepts and data long. And now we've added family equals binomial link equals logit. This is the part that tells it it's logistic regression. So my first model, I'm going top down here. I've got the moderation or the interaction. The next model I have just main effects. Now, if I fit these, how can I, oh, it's down there. How do we compare two models?
Log likelihood.
Likelihood ratio test. And here we go. So I'm going ANOVA with the two models. What does it tell us? No what?
Not better.
What's not better?
The more complicated model.
The more complicated model. Which model is more complicated? Model 1 or model 2?
The one with the interaction.
The interaction. So model 1 is more complicated. It's not better. So model 2 is our best model thus far. So model 2. Okay, so we can use, and again, I need to go back and change this to NitReg. So I'm going to put the two models together side by side in a TechReg table. Okay, so here's the interaction. What the Wald test gives no stars to this parameter estimate. But what about main effects? Is there a difference over time? And is there a difference by dose? Candace, you're nodding. Mike, you're nodding. Everybody nodding. Right? Carter. Now, wait a second. In this model, dose has no stars. But here, dose does have stars. What the heck? Where did the stars go?
Interaction sucked them up.
The interaction sucked it up. You cannot trust the stars on a main effect when the interaction is also in the model. So the likelihood ratio test told us we should not have that interaction. So we should look at the without the interaction model. And that says, yes, there is a dose effect. Yes, there is a time effect. Now, these betas are in terms of the logit scale. Going back to logistic regression. What do we have to do to get them? We have to do the extractor function, which is right here. So here I've now there in the TechReg helper function, there are three functions. There's extract GLM exponentiate, extract GEE exponentiate, and this one is extract GLM ER. You notice how the middle part of that function right here, you can't use the one that was just GLM. And the reason is I had to build custom functions for each type of model because of the way the output is packaged together. GLMs package the output different than GEE, different than GLM-ER. Okay, so once we do this extractor function, our parameters are no longer on the logit scale. They're now on the what scale? What's the exponentiated beta for logistic regression?
Odds ratio.
Odds ratio. Thank you, Jill. So now, what is this telling us about high? The high dose. Women on the high dose have 64% higher odds of experiencing amenorrhea at a given time. What does this time variable tell us? The odds of amenorrhea more than double each 3-month interval. Now, that's okay to talk about, but usually it's helpful to visualize, right? We like visualizations. So I'm going to go in and try to make a simpler plot syntax. This is the old syntax, but we get a picture like this. So what happens? So here we have the predicted probability of amenorrhea on the y-axis. On the x-axis, we have time that's going from 0, 1, 2, 3. And then we've got a line for high dose and a line for low dose. What happens to those chances? Over time, the chance goes up. And the two groups are, of the two groups, the high group is higher than the low group. But it's, now, in the logic scale, they would be parallel. In the probability, we see this curvature and they kind of get bigger, like the gap gets bigger. If we were to graph this on the logit scale, we would see two straight parallel lines on the logit scale. But when we exponentiate and go back to the probability, we get these curved lines that are not necessarily parallel. And here I kind of, let's see what I do here. This one I plotted from 0 to 100% and made a gray line at 50%. So if a woman was on the low dose, how long before half of them experience amenorrhea? They've got to get almost out to that time equals 3. But what about for the high group? It's closer after 2. So I put a dark gray line on the 50% because 50-50, that's kind of a nice reference. And often we use that reference, you know, how long until half, and it's a 50% chance for each woman or half of all women. Half of all women on the high dose will have amenorrhea by this point, which is just after the second, this would be 3 months, 6 months, so this might be 7 months. But the low dose is going to take longer for half the women to have that experience. Okay. So this one, I let it zoom in, so it's not going from 0 to 1. I let it choose the zones. Okay. Now, last thing, optimizers. So we're doing maximum likelihood, but again, it's maximizing the likelihood not by a closed form, but by kind of a different computer algorithm. So we can have the integrals being solved by approximation. And this is where SPSS dies, essentially, on a lot of models. There are lots of different optimizers. I don't know which one SPSS uses, but it's not the best one. And so that's why it fails to converge more often. Sometimes when you have failure to converge, you can choose another optimizer and it will work. Sometimes you can try all of them and none of them work. If you change the optimizer and it works, should you try another one? No. If any of them converge, then the ones that converge will always give you the same answer. So we have different optimizer. In the LME4, if we were doing lemur control before, now we're going to use gleamer control. So here's one. This is one way that you can change the optimizer, is change the number of cycles. So the default is less than 50. So you can change this number to like 100, 150. It will take longer for it to run, but it's more likely to converge if you increase the number of cycles that it does before it calls it quits. The other one, we saw Nelder-Mead before when we did the linear mixed effects model. So it's much the same, but now we make sure we say gleamer control, not lemur control. But we can switch to this Nelder-Mead optimizer. And then there's this Optimix one. I put these on this encyclopedia so that I can copy and paste them from this when I have problems. So this page will exist forever as long as I can help it, because I always come here to copy and paste these. And then there's all these various optimizers you can try. Now, one thing, we're looking at change over time here. It's change over time. If we put time into our model, where was our model? That model? Haha. When we put time in the model, this is linear time, linear in the logit scale. Sometimes we think that odds change in a non-linear way, that there's like a peak and then it plateaus, or maybe the odds increase quickly and then plateau, or maybe they takes a while for the increase to happen. If you think that's the case, you can try time squared to get the quadratic, but it's quadratic in the logit, not in the probability. So you have to keep that in mind. So in this example, I wanted to look at quadratic time. So the quadratic time in this one has quadratic time, time squared, is more complicated than linear time. So you're likely to have convergence problems if you try time squared. So here I'm doing time and time squared, dose with time and dose with time squared. Okay, say this a couple times. If you think that a variable moderates change over time and you have quadratic time, so I think that dose might affect change over time, but I also have time squared. Dose must interact with time and time squared. You cannot have dose interacting with time, but not time squared. If you have time and time squared both in the model, you have to have any interaction for both. If dose is going to moderate change over time and you have quadratic time, you have to have dose interacting with time and dose interacting with time squared. I'm calling this model 3A here, because before we had model 1 and model 2. Which one was better? I think it was model 2. So now I'm going to do a model 3, adding in this quadratic time. So this includes the dose by time squared interaction, and we have to do the lower level dose by time. 3B is the same thing, 3C. These are the same model, just fit with different optimizers, all these 3A, B, C, D. So I can do a likelihood ratio test. 2 was our best so far. Now we're going to do 3. Uh-oh, what do we see? Model 3 is better. Model 3 has dose interacting with time and time squared, and that's a much more complicated model. It has 6 parameters instead of just 4, and it turns out to be better. So here I'm going to make, here's to show you. I put models 3A, B, C, and D. All of these models are the same parameters. They just have different optimizers. Check out the parameters. What about the intercept? Are they identical? Pretty close. That first one's a little bit off. Let's go to the highest. Let's do this dose by time squared. If an optimizer converges, it converges to the right thing. You don't have to try another one, because say all of them converge, they will all converge to the same thing, the same solution. Check out the AIC, BICs. Are they the same? That first one's a little different, but the others are like identical. Again, if an optimizer works, you don't have to worry about trying anymore. So if the default converges, you don't get any errors, don't even look at it anymore. Okay, so here is when I plot that model. Does this tell the same story we had before?
Not exactly, because they don't run parallel. From what I remember before, you see a interesting sort of split in the middle there.
Yeah, so I think that this dotted line, the low dose, it's crossing the 50% threshold right before the last time point. But what it's done is this high group has risen more sharply at the beginning, and then it kind of plateaus a little bit. And so it's hitting the 50% line right after that third observation time point. Yeah. So is the story the same? Largely, but the estimates here are going to be more accurate because this more complex model fit the data better via testing it by likelihood ratio test. GLMMs. There's your first logistic multi-level model. Is that a lot to digest? So it's putting together everything we've done before. We've done fixed effects. We've done random effects. We've done time and we've done time squared. We've done logistic regression. Now we're just putting it all together. Now in the encyclopedia, I have one more binary example, and we'll work through this next class time and do a couple more slides on binary logistic multi-level model. And then there's one on count outcomes, and we'll do that a week from today. And I'll have some readings put up for you, but we will not do a homework assignment on these. And then the last week when I lecture, we'll do the sample size and power, and then you'll do your presentations. Okay, I'm okay if we want to wrap up early. I've gone over 5 minutes several times. We can end a little early today. I will definitely stay on for questioning though.
Thank you.
Welcome.
Can I ask a very quick question about today's lecture? In this model, quadratic model shows better model fit than linear model. I was wondering, when do we have to check the time effect? Like, when do we have to check the time effect? Like, after deciding the final model, and then we can check the input the linear and quadratic and then
yeah, I would
compare.
say that this one is my final model. Before I did 1 and 2, and I decided 2 was better. But then the reason that I wanted to look at quadratic time is based on the exploratory data. It looks like the blue columns got taller and then kind of stopped, and the red were like lagged and then they started catching up. And that's indicative of a quadratic effect, that the first one comes up and then the second one comes up after it. And so that's why I checked for the time squared by dose effect. Now, it kind of goes back to Joey, what he talked about last time, about how many things do you try? It should be theory-driven and data-driven both. It makes sense. Now, this is data that we don't care about. We didn't gather this data. But these researchers really thought that that high dose should kick in faster, but there's some women that it does nothing for. So it's not going to keep going forever. So it's going to kick in faster and then kind of plateau, where the low dose would maybe take longer, but eventually would catch up. And because the researchers knew that about this drug, that's what they wanted to test. So I'm just recreating an analysis that certain researchers ran and published. If this was me, I wouldn't have known that that's something that I should be interested in.
So we can check it first after checking the exploratory data analysis, and we grab the distribution of the data, and then we decided quadratic or linear model, right?
Mm-hmm, mm-hmm. Yeah. So the decision to try it should be based on theory and exploratory data. And then when we fit it, we can decide, does the data back up the linear or quadratic better? The main research question here is, does the dose change what's happening over time? It was specifically a dose by time interaction. When we did the dose by linear time, it was not significant. And that left the researchers scratching their head, which is why they tried dose by time and time squared to see if there was not just parallel differences, but arching differences.
I see. Thank you.
Good questions. See, Allison, Julie, Jill, Joey, other questions? I think Alex, you're staying behind for the data rodeo, right?
Yeah, I just had a question for the notes that you took during our consultation.
Yes.
No, I can't find those.
Okay, so bye Joey. The original notes, if you go to, remember you had to do like the quiz where you filled out information about it. If you go there, I uploaded a photo in my comments back to you.
Oh, to the quiz?
Yeah, to that quiz.
Okay, I will check there.
Let me see. So which one was that? Let me make sure excuse me, yours are there, Allison. I think I got everyone there. So it's in the consultation and determination. And then let's see if I gotta go, is speed greater to see them. So Allison, there you are. So yeah, so in my comments back to you, I uploaded a file that's a JPEG.
Okay, I missed that.
Yeah.
Okay.
And so let's see, here's yours. Yeah, we need to rotate that too. There, that's kind of, there it goes. It cleared up. Yeah, so there it is.
Okay.
So yeah, so for reaction time would be a lemur model, but your accuracy, whether they got it right or not, would be this gleamer model.
Yeah, and that's why I was like, okay, I need to remember, because I didn't write as much detail as you did in my notes. So
yeah. So yeah, did you find it on your end in Canvas?
I haven't.
If you don't, let me know and I can send it to you.
Yeah, I will do that.
And if you want to get together and chat over it and go over, like, try it in R, and then if you want me to go over it with you, definitely send me an email.
Yeah, that's what I was planning. I want to try it first and then talk to you. So that's what I'm planning on doing.
Awesome. Well, you won't have a homework assignment anymore, so you can do that.
Yes, thank you. It's very helpful.
Okay.
All right.
Okay. My kids are singing Frozen out there on the top of their lungs. Can you hear it?
No, I can't.
It's so noisy. It's great. I love it, but it's sure noisy. Let me see.
How old are they?
Okay, see, look, I have in here. See, I have the dates that go with every 3MS. So let me do this. Okay. Go to Okay, I am going to. Okay. Okay. So now I have, so with every 3MS, I have a date.
Oh, do you want this to still be recording?
Oh, thank you.
You're welcome.